So, welcome to Unknowns. Today, we are going to see how to choose a research topic. I think it's the right time to talk about it since many are entering into your fourth semester masters and also final semester in your PhD. So it's the right time to see how to choose a topic. And I'm very sure this will also be useful for fresh PhD students who are about to start a research. Okay, so it is very much important to acknowledge the fact that there are thousands of human beings in our area of research who has done the research before us. In mark of respect, it's very much important to read the research article in our field, on our chosen field. Or in other way of saying, either you choose a field first and then develop a research methodology, or you can see what is required for the society, and from there you can identify a research gap. Either way, this presentation I'm going to cover how to identify a research gap. Okay, so either you choose a research area of research. Say, for example, I'm being microbiologist. I will highlight you few of microbiology examples, and then I will see how to derive a research question and how to derive a methodology to solve the research question, which is basically a research that we do, and then the expected outcome. Okay, expected outcome is nothing but how to meet the research objective, and the success of research will be reflected with either research publication, patent, or starting your own enterprise in the area of your discovery. So, how to choose a research topic? I would say pen down your idea. Okay. First thing first, you write down your idea. Here I have given an example, okay, of a student penning down their idea. Okay, I understand that the student is interested in human skin microbiome here. Okay, and she want to identify what is a harmful bacteria or beneficial bacteria, and they want to design a drug to tackle it. Okay, so basically, when you write down your idea, you are expressing your understanding, and then you are trying to derive a research question out of it. I will tell you why research literature review in the research is very much important. Look how this idea is going to be refined because of literature reading. So from here on, I want you to read along with me. It will be like a demonstration. Okay. So human skin microbiome is what I did. I just copied that idea. I Google it. Okay, and I have many papers coming up. This is a paper, Skin Microbiota, a source of disease or defense. I choose this paper is even though it is published to more than nearly 14 years back. I still choose it because the student understanding here is the all bacteria in the skin causes disease. So this is the right paper I thought to read about it, and also it has received nearly 1,000 citations. So let's read the paper. How to read a paper? Every paper will have these subheadings. Okay, title of the paper, author list, abstract, introduction, methodology, result, discussion, conclusion, references. If you are looking for a research gap, I would say discussion is the area to look into it. I will demonstrate you how to see. So this is a paper in the Google search, skin microbiota, a source of disease or defense. So see, in the abstract, it's very much evident if you sample the skin, these are the common culturable bacteria you are supposed to get into a nutrient tag, especially when you're using nutrient tag. Okay. So if you are imagining that I will isolate organism from the skin and then study what are the species. This is the first indication that it has been done more than 1,000 times before you. So let's see where to contribute. What are the research gap to fill in instead of just repeating what is already the discovery that has done. If you want to know what is already has done, there is no other way other than to read the research article. So when you read the research article, this is the first knowledge gain you get that common vector that is being isolated. Okay, so I want to know what are the current trends in the area I choose. The if human skin microbiome is a area I choose, so I just enter human skin microbiome and identify new strains and other stuff. Okay, so here is human skin microbiome research, and it is published 3 years back. Okay, So what are the factors that affect human skin microbiome has been discussed here, and these are the, valuable knowledge I gained from reading the paper. I know now that human surfaces may cultivate nearly 10 power 3 to 10 power 4 CFUs per centimeter square of the skin. These are the common bacteria I will encounter. Both, actinobacteria will be of higher proportion, followed by firmicutes and proteobacteria, and even in anaerobic forms will be present in a human skin. And these are the fungal diversities I will encounter, and these are the viral diversities I will encounter if I go for sampling the human skin. Before reading the paper or before formulating a research, it is very much important to know what are the basic findings in the area of chosen research. So when I continue to read the paper, I can understand that the first thing that strike me is, see, the school student has derived an abstract based on the fact that all bacteria in the skin is harmful. So here is a proof it is not so. Even it is very clear that Staphylococcus epidermidis it's required for maintaining the health of a skin. Okay, so bacteria is actually used to maintain the health of the skin. Okay, so immediately an idea strike out. Why can't we make a skin probiotics like we have a gut probiotics, since bacteria are playing vital role in maintaining the health of the skin? The answer, instead of searching research article, I just went to e-com platforms to see is there any other products available. Yes, there are many products available that clearly uses bacteria like lactobacillus plantarum to maintain the skin. So it is very much important when you are browsing for a research gap, when you want to start a research, better find a research gap in the area. If you are not doing a literature survey, you will end up repeating whatever has been already done. So when I continue to read the article, I know this idea of skin bacterial serum is a failure because the product is already existing. Until otherwise, I have a better idea to improve the product. I don't want to get into that research area. So I continue to read the research article, and these are the various data I'm getting from the research article. Even now, I know from the research article that if I sample my head, what are the bacteria I'm supposed to get? If I sample my foot, what are the bacteria I will be usually getting? If I sample the dorsal or ventral surface of the palm or the foot, what is the bacterial concentration and what are the species expected? So many research studies have been gone into skin microbiome, and they have filled all these knowledges in the field. So I continue to read. Even I know now that from newborn to, from your deathbed, microbial flora tend to change. The composition is also changing here. The violet is purple, yellow color is actinobacterial composition, green is proteobacteria, blue is bacteroids, and others make the gray area. I also understand that a bacteria that is useful in one particular stage of life on the human being can become harmful later. Okay, so there is no defined role for human skin microbiome. Even a mother delivering a child through natural way and also through a surgery, the bacterial composition differs. Child skin microbiome differs based on the way a mother is delivering a child. Okay, so there are many more topics in the paper that I'm skipping for now because I just want to make a point that reading a research article in a chosen area is the first thing you need to do to gain a knowledge in the field. Then you find the research gap. It is very, unless until you read and gain the knowledge in the research field, you are not going to find the research gap. Why to find a research gap? Unless you are filling the research gap, your research is will not find an impact in the society. So you need to know where is the research gap. For that, you need to gain the knowledge in the research field. This is a quote I like whenever we do a research gap identification. Either you choose an area, passionate about it, find a research gap and do it, or just see what is required for the society and find a solution. Okay. Two ways to approach a research question. So now my area of research for in this given situation, it is very clear that I want to do a research in human skin microbiome. So I would say at this stage, when you decide your field, choose a Google Scholar, not just Google. Okay. So in Google Scholar, I have Google human skin microbiome. In here, this slide, you will see anytime choose since 2024 so that you will get a latest research article. While finding research gap, it is very much important you read a latest research article because um, the research gap or the research question mentioned in the discussion of article in 2008 might have been solved in 2018 without knowing that if you are selecting a research gap from 2008 article, you may end up repeating the same thing that others have repeated. Okay, so just chronologically select the latest article. I will select 2023. Okay, when this is selected, here, since this search is done last year, I will use this example. So an update on current understanding of infant skin microbiome and their research challenge. So current understanding of infant skin microbiome. I like the words because I want to know what is happening in the field. So I will select this article. So I'm going to click here. So an update on current understanding, it is published on October 2023. Okay. Now I will get into the article. I will read the article. I will see that I have seen the graphical abstract. I have started reading the article. Okay, see, for writing one paragraph, 17 references are used. That's a hallmark of quality articles. Okay, each and every line will be supported with the research proof through a research article. So the first question I encountered while reading is, I was really surprised to see there is no efficient protocol for extracting high quality DNA from for shotgun metagenomics from skin samples. Okay, but logic is understandable because it's not a nutrient agar. The skin is has its own chemistry. The number of bacterial biomass in the skin is relatively lesser compared to the gut surfaces. So this is the first thing. So here is a choice. We can develop a protocol for extracting high quality DNA from the skin samples. Okay, then how everyone is you focusing on bacteriobiome of a skin, but the start we are it's very much clear from metagenomics work that archaeobiome and microbiome are also constituting profound composition in the skin microbiome. So studying their archaeomicrobiome and microbiome, fungal microbiome is also very equally important. And I'm also seeing how these bacteria, archaea, fungi, and viruses in the skin interact together. Is the studies in this area is also very poor. So this is a potential area to work on. Then finally, I was really surprised even in 2023, we do not know how to distinguish a skin related disease, the skin disease and skin disorder. The disease abnormality due to change in physiology or disease abnormality due to the infection caused by microorganism cannot be differentiated even by the easily differentiated even by the skin specialist. So it was the dermatologist, they are suffering to, this is a potential area for contributing to the area of dermatology. So the question can be framed from here. And then how these, everyone is just studying either culture-based bacterial diversity or they are studying on metagenomics or eDNA sampling and studying from there. But there is no integrated approach, integrating all the methods to have a comprehensive understanding of what's happening with the skin microbiome. So see, instead of just writing our own, penning down the idea, how was the idea here? And here we have 5 potential questions that needs to be answered in the skin microbiome research. So that's why I say it's very much important to read the article, especially when you do a literature search. In the discussion part, author usually describe about what was the, when you take a recent article, the author will be describing in the discussion about usually last 3 paragraph discussion is very much important. If you look into it, you can see that author will be describing about what was the problem in the field when he started the research, how he solved the research, and what are the research or what are the research gap that is yet to be solved. That's the third point. What I said is the potential we are looking for in the literature. From there, if you develop the research, it will be, it will be impactful research. I'm very sure you can either you will find a good journal for publishing the data if you fill the gap, and also you can start an enterprise like making a product out of it. I'm very sure in 10 years time you may become a unicorn from there. Okay, so I have derived 5 questions now. Before working on the questions, I want to check whether these questions are still live. Okay, this is from October 2023 article. So the first question, efficient protocol is lacking. Is it so? I want to look into Google Scholar and Google. This is the first paper I got. An efficient method for high molecular weight bacterial DNA extraction suitable for shotgun metagenomics from skin swabs. So this author has already started working and published an article. It was also closer to October period. They have published this article. So, should I drop the question? Not necessarily. I went in and read through the article, and here also author gave some hints how to develop the methodology. Like I said, if you look into the last two paragraph or three paragraph of discussion, they will talk about, we did not seek to benchmark the extraction, but it will be done in future to refine a method. So if you can prepare a DNA isolation kit, okay, as a commercial product, and develop on their methodology. They have used six methods and to check what methodology is yielding more DNA per sample. So if you can launch a kit or you can fine tune their protocol, still we can fill in their methodology. So likewise, we can cross verify the chosen area and check whether it's really worthwhile to start a research in that area. I would also suggest, this is an example I'm giving. You can also have an account in ResearchGate, that is a social media platform that will connect with the scientists. So if you really want to work on a particular research gap, you can select the specialist in the area. You can communicate with them. You can decide whether to, continue to work in that area, and they may also end up in refining your research question, which will be of immense help to you. This is an SMS or this is a WhatsApp message I want to share. It's very much important to have a research publication if you want to go to a European country or other, if you want to go to a high profile country, US or Canada, to pursue your PhD or post PhD. It's very much important to have a research publication. If you don't have a research publication, those universities will not encourage you to enroll in them. If you want to, I feel PhD and postdocs are harvest period for researchers, and where they can get a high salary, and it's very much rewarding. So this is a message I got from one of the scientists rejecting the applicant from an India, saying that it's not said openly. I know that because the applicant did not have a research article after her MSc, she was not denied PhD admission. So the research guides expect you to have a publication, because when they are paying you high, they want to make sure that their investments will turn into a meaningful contribution to the society. So to sum up, these are the four important steps you need to follow while identifying a research gap. Pen down your idea, and from there you can refine it, or if you are smart enough, you can find what society needs the most through, for example, from the microbiology point of view, and you can work on a research question. To answer the question, you can dedicate your dissertation period. So here I was trying to talk about plagiarism. So usually I find these two kinds of mindset. Okay, I have a lot of topics in mind. I don't know where to start. Start somewhere. That's why I say pen down your idea. If you have 10 ideas, pen down all 10 ideas. See which idea to prioritize the idea. Work on top three. Let's see what kind of research question you will come out with. Okay. I'm not understanding the research article that I'm reading. Yeah, this is a common, again, I used to see with MSc students. That's fine. If you are not, when you start reading from top to bottom, that is a 5 or 6 years of work represented in 6 pages. Okay. So if you are not understanding from first attempt, that's okay. Do not stay on a particular point where you are not understanding. Just keep moving on. Okay, when you're done with the entire research article, come back, read again from the first to last. Maybe in the first attempt, you may understand 10%. When you repeatedly reading the article, I'm very sure you will get it. That's why research gates are for. The plan B is you communicate with a particular author. If you have any clarification, you can also directly message them. But do not message them just by reading one time. Okay? 10 to 15 times read the article, increase the percentage of understanding, and narrow down your question. And then contact with the scientists. Definitely they will be very much helpful to, because that's why they publish. Okay, they want people to read their article and understand it, and they want to contribute the society through their research publications. When you ask about their research publication, they'll be very much happy to clarify your doubts. So this is the typical graphical thing I want to show. When there is a complex knots, fill them one by one. I'm very sure you will understand, end up understanding everything in the article. Okay. So regarding literature search, I will do a separate video. I hope this video will be useful for you. Thank you for watching. Notes.